Hi, I'm an academic clinician, and today I want to share with you the development of the MenDeVar index. Six years ago, we developed a rapid, scalable method for analysing the genetic diversity of the 4 peptide antigens in the meningococcal B protein-based vaccines Bexsero and Trumember. Similar to MLST typing, each novel amino acid peptide sequence was assigned a new variant number, and the combination of these was assigned a Bexsero antigen sequence type, as seen here for the vaccine antigens. The BAST and antigenic profiles are available for all isolates in the MLST database, or by querying your genomic data on the website. The peptide variants present in this isolate, or absent as in the case of NAD-A, can be seen here, along with the BAST number. Although BAST typing allowed large-scale genomic surveillance of antigenic variants, there were no rapid or widely available methods to assess protein expression or cross-reactivity of these antigens. This is of particular importance when assessing the applicability of the available vaccines to a given disease-causing isolate. As you can see, for this isolate, the 2 vaccines have differing reactivity for both Bexsero and Trumember. We therefore decided to develop the meningococcal deduced vaccine antigen reactivity index to address this gap. MenDeVar index was developed to combine complex genotypic and phenotypic information into a simple, evidence-based output for use by frontline healthcare professionals. First, we performed a literature search to identify all the laboratory tested isolates with details about each peptide variant they contained and their reactivity in phenotypic testing, that is the meningococcal antigen typing system for Bexsero, and the measure assay and serum bactericidal activity assays for Trumember. This algorithm shown here was then used to assess each peptide variant which has been tested more than 5 times in these laboratory assays. For each variant, the phenotypic data was combined to establish how each peptide variant reacted to vaccine-derived antigens. If at least 3/4 of those tested were reactive to the vaccine antigens, then the variant was considered cross-reactive. If less than 1/4 of those tested were reactive to the vaccine antigens, then the variant was considered not reactive. If between 1/4 and 3/4 of those tested were reactive, then we deem there to be insufficient data to categorically assign these to be red or amber. All the peptide antigens that have been assessed to date were assigned green, amber, red, or grey. Then, for a given meningococcal isolate, the antigen requirement shown here can be used to define its overall MenDeVar index. That is, if it possesses at least one of the green antigens, then it is assigned green. If there are no green antigens and it contains at least one amber antigen, then the isolate will be amber. If the isolate possesses all red antigens, it will be red. And for any other combination, the isolate is assigned grey. The index will be annually updated to accommodate new literature. The visual output of this information can be seen as a traffic light output for each vaccine, listed with the publications that support this output and the caveats. The caveats are provided to allow users to understand that this is a deduced index with data from multiple sources, with definitions of cross-reactivity that are considered measured but pragmatic, and from studies with participants of varying ages. As many reference laboratories are now sequencing meningococci in real time, this information can be available hours after sequence data are generated. Users can analyse their genomes by uploading them to the database, or by using straightforward plugin for the MenDeVar index from the MLST homepage by copying and pasting PCR products or uploading FASTA files of genomes. I thank my colleagues in genomics, microbiology, and public health in both Oxford and Scotland. Thank you.